Okay, so welcome back, everyone. My name is Adam Stieg. For those of you who don't know me, I'm the Executive Director of BioPACIFIC MIP here at UCLA. And I'm going to be emceeing the next two sessions, which are really exciting talks that I'm looking forward to. The first is by my colleague and friend, Tang, who has been at UCLA. I guess I realized the other day when I was looking at your CV that you were here for 15 years before we ever met.
Yes.
But it's been a pleasure in the last few years with this BioPACIFIC activity, getting to work with you and know you about all the exciting things that you're doing in your lab and what it is that we're hoping to do in the Living Biofoundry. So Yi has come to us, as I said, 2004. You know, he did his undergrad chemical engineering out at Penn State before he did a PhD at Caltech, moved out to California and found the reason we all like to live out here, and then ended up at Stanford before coming down to UCLA to set up his own lab. And so he's got, you know, litanies of awards and fantastic papers that I won't go through. But I think you'll find this this lecture informative in the sense that what we're hoping for is that you can sort of shine a spotlight on what synthetic biology is, what it can do, give you new ideas about how you might be able to utilize it in your research, and hopefully come to work with us here at BioPACIFIC MIP at the Living Biofoundry. So without belaboring the intro too long, I just turn it over to Yi, and we'll look forward to your talk for the next, I guess, 40 minutes or so.
Okay, thank you very much, Adam. Can you guys see the slides?
It's in the slide. Yeah, there you go.
Okay, thank you. Well, I'm really happy to be here and being part of the team that applied for this this funding. I'm just super happy that it's funded and we're here, the first summer school, to tell you about what we do. And hopefully my presentation today will give some background on what I think is synthetic biology and what are the capabilities of synthetic biology and how it relates to the mission of BioPACIFIC. So I will give an overview of synthetic biology. Actually, I don't have a single slide on my own research here, which is kind of which is not as exciting as an overview. But I'll talk about what synthetic biology is, and also just briefly introduce the BioPACIFIC capabilities. So my outline is I'll give an overview, and then I will talk about three areas where synthetic biology has played a major role in chemical synthesis and highlight some of the accomplishments, in my opinion, are the milestone advances in the field. And then I'll conclude by talking about the biofoundry capabilities. And that will lead into our next speaker, Colin Harvey from Hexagon Biosciences. So the question, of course, is what is synthetic biology? This is a term that you started hearing about, I'd say, roughly 10 years ago, and this has really taken off in the mainstream science as well as popular science. Yet this name is incites some confusion. You know, what is synthetic biology and how is it different from what we already know about genetic engineering or other types of biotechnology? So I just want to offer a few people's thoughts and my own thought on this before I dive into the what I think are the success stories in synthetic biology. Synthetic biology, to me, is an umbrella term that refers to engineering of living organisms towards new functions and properties using fundamental biological principles. And by umbrella, and I mean it covers a lot of the old classic discoveries and technologies, as well as encompassing new technologies that are continuously changing the field of biology. And it's at the interface of biology and many disciplines, as you will see later. A good analogy of this can go back 100 years to the invention of the term synthetic chemistry, which we all accept now as a core discipline in chemistry. You know, synthetic chemistry is really to take what you learned about chemical principles, bonding, reactivities, and apply it towards making different molecules that exist in nature or completely new, that are small molecules or polymers or even macromolecules. So synthetic biology is very much the same way, is that it takes core biological principles and apply it towards making something useful or doing something beneficial to humankind. It covers some of the existing technologies, such as transgenic plants, recombinant DNA, and also include new technologies, which I'll highlight some in the coming slides. This is based on the fact that we can have access to blueprints or genomes of many organisms much more readily and have increasing tools to engineer them for different applications. So synthetic biology has been compared to a bacterial cell with a integrated circuit inside, and I think that's a very appropriate comparison. So here are some more definitions of synthetic biology, which I think can help we all appreciate this emerging field. And as I mentioned, synthetic biology is seen very differently by biologists and by engineers and by chemists. But it's really definitely coming on its own as a discipline. So generally, synthetic biology, according to this review, which I think is very well written, is the use of molecular biology tools and techniques to forward engineer cellular behavior. Another definition is that synthetic biology is the application of science, technology, and engineering to facilitate and accelerate the design, manufacture, or modification of genetic materials in living organisms. This is by a committee in the EU. Here is in the Nature Review Microbiology paper, here's a timeline that covered up to 2014, and of course, many have since happened since 2014. And you'll see that, as I mentioned, this encompasses many of the old technologies dating all the way back to 1960, and including the discovery of the lac operon and many of the classic things that we learn in genetics or biochemistry textbooks. So you might want to ask, why is the synthetic biology any different than what we already know? Biology has been engineered to produce useful things for a long time, right? Since like insulin and human growth hormone and other human therapeutics. So why in the 2010s this synthetic biology term really emerged and is taking over this part of biotechnology? And I think a better definition of synthetic biology comes from our North California neighbor in the Simberg Center at Berkeley. And I'm not going to read the whole quote, but you can see in red, I think that really differentiates synthetic biology from traditional biotechnology, or not differentiate, maybe evolved from traditional biotechnology, is that the synthetic biology has the element that distinguished from traditional molecular biology is the focus on the design and construction of core components, parts of enzymes, genetic circuits, metabolic pathways that can be modeled, understood, and tuned to meet specific performance criteria, and the assembly of these smaller parts devices into large integrated systems that solve specific problems. Okay, so if you compare this to electrical engineering, where you design circuit board, you have something objective in mind, then you model the circuit board, then you combine different standard parts together into an integrated circuit, and you go through the design, test, learn cycle to optimize it to achieve what you want to do. And I think that's the essence of synthetic biology, is to incorporate the design, assemble, test phase to have biology do useful things. And some of the core concepts, just to elaborate from the previous slide, is that there's abstraction hierarchy to make very complex operations simplified individual steps, and that can be worked independently at each level. So you start with DNA, you assemble the parts, and then you assemble the devices, and you integrate them into a system in the engineered microbe or engineered organism. There's a concept of modularity, right? Again, going back to integrated circuits or even synthetic chemistry, is to use individual components that can operate independently as part of a large system, and you put them together in a very complex way to achieve certain objectives. Standardization is very important for any engineering project, is to establish uniform criteria, parts, components, or materials that can directly affect the design of a product, right? So you want something to be controlled, you want, or to be induced by light, you have a standard part that you can take and incorporate into your system, etc. And then something that very intimately related to BioPACIFIC is automated construction, is that once you have these standard parts and modules, the ability to design a complex system and then order it without the need for traditional molecular cloning techniques, is to integrate automated operations, incorporating these standard parts to construct to accomplish an objective. So I think these are very important distinctions of synthetic biology from traditional technologies, and that's what makes it exciting and makes it a it's becoming a very popular for engineering disciplines to jump into biology, applying engineering principles. Some of these are outlined here. So synthetic biology is fueled by new technologies, I mentioned. So if we look at the central dogma that you find in textbooks, of course, you go from DNA replication, and then can be transcribed into RNA, then translated into proteins, and then also incorporate catalysis by proteins to make metabolites. In our case, we're interested in making monomeric compounds for polymerization. And I highlight some of the technologies that have really changed the way we think about biology and enabled synthetic biology advances. In the DNA replication area, or in DNA replication, we have the now old traditional recombinant DNA technology, but incorporating next-gen sequencing, CRISPR-Cas9 editing, and low-cost DNA synthesis. In RNA, you have transcriptomics, ribozymes. Protein, you have directed evolution, protein design, and natural amino acids that can be incorporated into proteins. And then finally, in metabolites, you can incorporate metabolic engineering, cell-free synthesis, etc. And all these new technologies are fueling the rapid development in synthetic biology. I just want to highlight 3 technologies, many of which are well known to the audience, but I think we have to mention them for the synthetic biology era, and I think perhaps Colin will follow up on some of these in Hexagon as well. The first, of course, is DNA sequencing, right? So in order to engineer something, we must know its blueprint, and we must understand what's already there. And sequencing enabled by next-gen sequencing technologies has really dramatically lowered the cost of DNA sequencing. And this is a chart by NIH, and this is a widely searchable chart. You can find it anywhere online, and it just shows you the dramatic decrease in sequencing costs from as short as 20 years ago, from $10,000 per megabase of DNA to essentially free now in 2021. And it's marked by several developments, big drops in sequencing technology. So now we can sequence basically any microbe, any plant, any species relatively fast and much, much cheaper. And once you have the sequencing, you want to alter the blueprint, you have to make DNA. And then another falling cost is the price per DNA letter, or about the cost to make DNA. Synthetic DNA by companies like Twist or others. As you can see, again, in 20 years, the cost of making DNA per DNA letter has dropped again significantly, over 3 orders of magnitude, close to 4 orders of magnitude in cost. So now you can, and the ability to make longer synthetic DNA has also increased dramatically. So our ability to make any kind of genetic building block has been significantly enhanced compared to just 20 years ago. So again, synthetic biology relies on the assembly of these synthetic genetic building blocks as standard parts. So this is a very, very important development as well. And two more technologies I just want to highlight, but think everybody's in science knows. One is, of course, genome editing, is that now you can very simply customize the chassis or the microorganism or the species that you plan to engineer by precisely introduce edits or deletions or insertions or mutations. And this, of course, was a Nobel Prize in 2020, as well as seminal contributions by Fenjiang, MIT, to allow genome edits on very complex species. So you can now take any organism that you like and then can precisely making changes to your liking to achieve a certain objective. And the last advance that I think really impacted the synthetic biology is directed evolution. Of course, this was a Nobel Prize in 2018, highlighted by Frances Arnold at Caltech. She was my committee member at Caltech, and she was, at that time, she was able to provide me a slide on directed evolution. This was 20 years ago. But I think it's more fitting than ever to use this slide because it just really shows the concept of you take a gene, you mutate it, you select for the improved mutations, and you iterate this process in what's known as directed evolution to evolve the function of a natural protein into something completely different. So this allows new functionalities to be introduced into proteins, introduced into cells. And now if you combine these new technologies together, the sequencing, the DNA synthesis, the editing, the protein evolution, you can imagine that we can get these microorganisms, living organisms, to do many new things that were never thought possible. So having these in mind, I'd like to talk about some of the applications I thought are really classics in the field. And by classics, I don't mean old. I mean, they're still relatively new accomplishments, but are really a milestone and demonstrating what synthetic biology can do, especially towards chemical synthesis. There are many other technologies such as cancer immunotherapy and others, which I'm not including here because it's not directly related to the mission of the BioPACIFIC. So one of the first applications of synthetic biology, I think in my recollection, synthetic biology, the term really emerged was the biofuel explosion in the late 2000s. So the biofuel idea is to take solar energy and get the sugars out of the plants that thrive on solar energy, and then convert the sugars using engineer microorganisms to make biofuels, including compounds like methane, alcohols, biodiesel, etc. And to do it in a very renewable way that we no longer rely on petroleum. Of course, this is not a new science at all. Biofuels have been around forever. Of course, people know that yeast can make ethanol to make biofuels and other liquids for entertainment. And so how yeast do it is they take sugar, they perform fermentation under anaerobic conditions, and that will generate ethanol plus carbon dioxide. So this has been around forever, but I think the impact in synthetic biology is to do this better, and as well as to make new biofuels. Right? So if we look at a challenge perhaps in the late 2000s is how do we make other kind of short chain alcohols like 1-propanol, 1-isobutanol, butanol, methylbutanol, etc. These alcohols, because they have more hydrocarbon content, have higher energy density, as well as lower hygroscopicity, which means that they don't absorb water as much as ethanol. So these can be think of as better fuels, but they're not always made by microorganisms. Some of them are, but most of them are completely foreign to the microorganism. So how do we engineer, for example, E. coli to produce these next generation biofuels? This was accomplished by my friend and colleague James Liao when he was still at UCLA. So this is a UCLA accomplishment. He's now the president at Academic Sinica in Taiwan, and also by his postdoc Shota Asumi at that time, but now he's a professor at UC Davis. They came up with an idea is that if we can introduce two new enzymes into E. coli, one enzyme is 2-keto acid decarboxylase, followed by alcohol dehydrogenase, then we can take these keto acids shown above, which are actually quite common metabolites in E. coli that are en route to amino acids, and turn them into these short chain alcohols. So the keto acid decarboxylase will decarboxylate these keto acids to generate the aldehyde, and then followed by a reduction by alcohol dehydrogenase to give the alcohol. So this was accomplished in 2008. They designed their pathway and they searched for gene candidates from different organisms, and they built their strains to incorporate the KDC and ADH. And they put into different E. coli hosts that are high-producing hosts of amino acids. So these hosts are naturally can produce a lot of these keto acids. And they tested these strains, and they were able to make very high quantities of these alcohols, especially isobutanol. This was in 2012, 2010, won the EPA Presidential Green Chemistry Award for ability to make these important biofuel molecules from a very common E. coli organism. And this led to the startup company called Gevo that commercialized this technology. So I think this is a very, and looking back now, this is a quite simple approach, right? Just two enzymes into E. coli, but at that time, I think this really demonstrated what synthetic biology can do in transforming a common microorganism into a biofuel producer. Another example followed in 2013 is the production of gasoline by E. coli. So gasoline, of course, is short chain alkanes, C8 to C10 alkanes. These are definitely not found in microbes. So this group in Korea by Sangya Police Group, they engineered E. coli to produce short chain alkanes in reasonably high quantities. So what they did a series of things, modifications to the host. First, they disable fatty acid oxidation. So in this particular step, the enzyme FAT-E, which introduces unsaturation into fatty acids that makes human unsaturated fatty acids, is disabled. So this prevents the oxidation of fatty acids. They disabled a regulatory protein that limits the amount of fatty acids that can be made in cells. So this really upregulated fatty acid production. They also engineered, using protein engineering evolution methods, to engineer a thioesterase that can stop the growth of these fatty acids at the C8 to C11 stage. The most common fatty acids in microorganisms is C16, but that's not desired to be a gasoline. So they engineered this with mutation to limit size to C8 to C11. And then finally, they introduced reductase and decarbonylase from other organisms into the E. coli to take the fatty iso-CoA into fatty aldehyde and finally into short chain alkanes. So this is an excellent example of multiple modifications to the host, both deletion and insertion of new genes to make a gasoline molecule. Okay, so now I want to move to chemicals. So I just mentioned biofuels, which is of course also chemical. By chemical here, I mean commodity or value-added building block chemicals. And synthetic biology offers a powerful and green platform to produce these commodities and can be used to replace petroleum sources, which are the source of these molecules before. Many petroleum-derived chemicals, such as solvents, monomers, fragrances, can now be produced using engineered microorganisms, some of which are shown here on the right. The process of chemical synthesis many of these chemicals are not natural to the host itself, so it will require extensive use of the design, build, test, learn cycle, as I mentioned before, for synthetic biology, and must ultimately be able to be cost competitive with traditional chemical routes. So just keep in mind, just because you can make the compound doesn't mean it will be commercially viable if you cannot compete in cost with petroleum-derived chemicals. So there's a lot of design, a lot of testing, a lot of optimization involved in making these chemicals. So I want to show you three examples, which I think are three examples that are really the milestones in the field. The first is a monomer for making Sorona, which is a fiber made by DuPont. Sorona is a copolymer of terephthalic acid and 1,3-propanediol. Okay, so this is 1,3-propanediol, and here is the 1,3-propanediol monomer unit in the copolymer. This is a very strong fiber resistant to stains, and it's used by DuPont to make carpets or rain-resistant apparels. And I'm sure some odd people in the audience are probably have clothing derived from Sorona. So Sorona was previously, this copolymer was previously all made by petroleum-derived sources. But in the 2000s, DuPont decided to embark on an effort to replace some of the core monomers in 1,3-propanediol by a microorganism or derived source. So what they did is they engineered E. coli to make this 1,3-propanediol and then copolymerize it with terephthalic acid to make Sorona. There's also efforts, successful efforts to make terephthalic acid from microbes, but that's still, that's not as scalable as 1,3-propanediol yet, as far as I know. There's also one that used Presidential Green Chemistry Award. And this process actually took a long time to optimize. As you can imagine, you need to make this very cheaply compared to the petroleum source. So this involved, as far as I know, a decade of engineering and optimization at a company like DuPont to make it economically viable. So they performed this in E. coli. This base string of E. coli has no capacity to make propanediol, even though propanediol is known to be made by certain organisms, but E. coli does not make it. After engineering a multitude of deletions, evolution, and new genes, they can make 130 grams of propanediol from glucose, which is more than 50% yield, carbon yield. And they balance this string so that it's between growth and production of the chemical, so that you don't sacrifice microbial growth, and also you achieve maximum possible yield. The way they did it is quite Herculean, in my opinion, if you read the papers. First, they replace how glucose is uptake and phosphorylated inside the cell so that to make it more efficient. And then they routed glucose through this dihydroxyacetone phosphate pathway, DHAP, and introduced new enzymes into E. coli to turn that into glycerol. And then they removed the extra hydroxyl group in glycerol using two additional enzymes that's introduced to make the 1,3-propanediol. These genes are mined from different organisms, introducing to E. coli, and followed by significant optimization of their activities to accomplish this. Another chemical related to 1,3-propanediol is 1,4-butanediol. So it's just what contains one additional methylene group. This chemical is never been found to be produced by any microorganisms. It's used as solvent and as a monomer for plastics. Large amount is converted to tetrahydrofuran to make fibers such as those in spandex. Annual production of 2.5 million tons, and only from petroleum-based feedstocks before this accomplishment by a company called Genomatica down in San Diego. There's no known microbial pathway, so the pathway must be completely designed and optimized. So what Genomatica did here is they used a computational tool to design the pathway based on known enzymes with known reactivities. So this is sort of like building a circuit, integrated circuit analogy in electrical engineering. And then they sourced these different enzymes using synthetic DNA, combined them, put them into E. coli, tuned their expression, eliminated site competing pathways, eliminated bottlenecks, and then finally it used the pathway shown here, starting from succinate, which is a central metabolite. They were able to get about 20 grams per liter of butanediol. And this process now has been scaled up to be commercialized. So this is a great example of something that's not known in nature, but is able to be engineered artificially using synthetic biology. One more example, more recent, from a group in Manchester, in UK, Manchester Center of Synthetic Biology for Fine and Specialty Chemicals. What they did here is they engineered E. coli to make all kinds of different aromatic monomers for polymerization. I'm not going into details of what they did, but you can see here I boxed some of the metabolites that are intriguing to use as monomers and also relevant to BioPACIFIC's, some of BioPACIFIC's goals. For example, they're able to make styrene at a reasonable titer for vinyl phenol and as well as some other molecules. So this illustrates the potential of rerouting metabolism to generate all kinds of natural monomers from E. coli. Okay, just a quick side on chemicals is the food market. Synthetic biology has also made a significant impact on replacing traditional food sources. We all know about the artificial meat, but the one example here I want to show is the artificial milk. So this company, Perfect Day, so some of you might have seen their products in the supermarket. They have ice creams derived from milk proteins produced using synthetic biology. So instead of relying on cows to make milk, which requires a lot of land, a lot of water, they emit a lot of gas, they cost a lot of energy, we can, the Perfect Day did is they engineered the yeast, dairy yeast, to produce a milk protein, artificial milk protein, and derived from sugar. And they can optimize this process, achieve high density fermentation of the dairy yeast to make the milk protein, and then formulate it to make the very same milk proteins that the cows can make. And again, this product has already gone into dairy products such as ice cream in the supermarket. And I have no affiliation with them, so I'm just citing this as a great example of synthetic biology. Okay, so lastly, as an example, I want to talk about synthetic biology for medicine. And this is something, again, our next speaker, Colin Harvey, will elaborate on. Medicine, is, as you'll see, can come in two things, especially relevant to synthetic biology. One is to the discovery of compounds known as natural products and to produce them. The other is to engineer T cells for cancer immunotherapy, which I'm not going to talk about. So I'll briefly introduce natural products, show again some of the landmark accomplishments, and then I'll let Colin take the rest away. Natural products, just for those people that don't know, are complex molecules produced by nature that turn out to be therapeutically important. They are produced in nature mainly as a chemical warfare between microbes or between plants and microbes and plants, but they can be harvested by human beings to treat different diseases. So some of the molecules that are very famous in the antibiotic field, erythromycin, tetracyclines, penicillins, are all known as natural products. Also, plants make natural products, again, as a self-defense mostly, such as artemisinin, which I'll talk about in a second, and as well as Taxol, Artemisinin, again, is another Nobel Prize in 2017 for treating malaria. Natural products has a lot of important pharmacological activities, including the antibiotics, cholesterol lowering, immunosuppressive, anti-cancer, and constitute a major source, even to this day, of drug discovery and also inspirations for synthetic drugs. And more relevant than ever, natural products are mind-bending compounds or mind-smoothing compounds, such as some of the compounds listed here. And caffeine, of course, is one of the oldest natural products consumed by human, but there are also other natural products that can significantly alter human behavior. What can synthetic biology do to produce medicine? So in nature, for example, take opium poppy that produces morphine. Nature uses a series of enzymes to construct simple building blocks into very complex structures in what's known as a biosynthetic pathway. But what synthetic biology can do is that we can take these enzymes pathways and put them into fast-growing microbes such as yeast to make the molecule from the microbe instead of relying on growing plants, which can take significantly longer. And what we can also do is that we can then engineer these microbes using new additions of genes, evolved genes, and deleted genes to make novel structures have better activities that's not observed in nature. So this offers a faster growing, genetically more manipulable source to these very important compounds. I would say also coincided with the synthetic biology era is the production of artemisinin precursor using synthetic biology. Artemisinin is this endoperoxide containing diterpene, sesquiterpene that's discovered from the plant of Artemisinium annua, and has been a primary, that's been one of the most effective chemicals for treating malaria. And it's always being extracted from plants, so it's the price fluctuates depending on how well the plant grows, but the demand is projected to be very high. This is outdated projection I've shown here, but the projection is very high for this compound. So if there's a reliable way of obtaining this compound without relying on plant growth, this will be optimal for producing this drug, especially for the African subcontinent. So in 2013, Jay Kiesling, actually it should be 2003, not 2013. 2003, Jay Kiesling's lab at UC Berkeley published the first report on taking the artemisinin pathway from the plant and transplant it into yeast and allow yeast to make a compound called artemisinic acid, which has the decalin scaffold that you see also present in the final product. They were able to, following a series of optimizations and a series of papers in the next decade, they were able to take this compound to produce at 25 to 30 grams per liter. This compound then can be very easily semi-synthetically turned into artemisinin in a few steps. This technology translated into one of the synthetic biology powerhouse companies, Emmerus, in the Bay Area. And now this technology is used widely. It has been licensed by Sanofi to produce artemisinin using this fermentation followed by semi-synthetic method. One more example of synthetic biology to medicine is making morphine or opium poppy natural products. And these are, of course, painkillers. We all know the problems of these compounds, but they are still very effective as painkillers. The total synthesis of these molecules being accomplished, but there's very many steps and not very practical in terms of scale up. So we still rely on isolating from poppy seed to produce compounds like morphine and codeine. The drawback, of course, is that many of these producers are genetically very inaccessible, and they're low abundance, so you have to have large fields of growing these poppy seeds, and they're crop dependent, again, climate, location, etc. So if we can somehow turn yeast cells or other microorganisms to produce these compounds and then grow them in a bioreactor, we might be able to achieve production of opium of these painkillers, opiates, in 3 to 5 days compared to over a year using the traditional method of isolating from poppy and scaling up that process. And this was accomplished by Christina Smokey's lab in 2015. They introduced genes. They sourced genes from all kinds of different species, including rat, bacteria, and other plants, and integrate them into the yeast cells in a 23-step biosynthetic pathway to make the molecule morphine and codeine and other opiate drugs. And this technology has translated into the company called Anthea, and which is also in the Bay Area, to commercialize the production of these opioids from microbial sources, as well as other analogs that might not be as addictive as the one we now know. Lastly, with the mind-altering compounds, Jay Keesing's lab, again in about 2018, published this paper on transplanting the cannabinoid biosynthetic pathway into yeast to make precursors. This, of course, we all know is a huge market, although their titers are still low, but this has led to the startup company called Dimitrex that can make compounds such as THC and CBD. So this, again, is one way of taking the joy of growing these plants from people and produce them in a microorganism instead. Okay, so coming up next, Hexagon Biosciences scientist, Dr. Colin Harvey, so founder, Dr. Colin Harvey, will talk about Hexagon's efforts to turn DNA into medicines using synthetic biology approaches. So in the last couple of minutes, I just want to, for those of you not familiar, just quickly introduce you to our efforts at BioPACIFIC using the Living Foundry to perform synthetic biology and integrate that with material synthesis. So as I mentioned, synthetic biology is somewhat a long process to go from DNA to metabolites, involving a lot of new technologies. And one of the key things I mentioned early in my introduction is that automation is key to assemble these parts together to perform synthetic biology. And that has many advantages over traditional molecular biology techniques. So here at the CNSI at UCLA, together with the CNSI at UCSB, we have acquired a state-of-the-art synthetic biology spinnaker system. This is actually a real picture now of the system installed at CNSI on the second floor. And this facility is run by Dr. Mike Lake, who will have a virtual instrumentation office hour tomorrow, if I believe. This system contains a automated robotics that traverses through a track in the middle of this platform and sample different instruments, including this liquid handling instrument here, to allow all these molecular biology techniques to be done in automation. This was installed in late May, and we've just performed our first successful run of this last 2 weeks ago, if I remember correctly, to perform some molecular biology techniques. So we're looking forward to adopting these systems, this system, along with other existing infrastructure such as genomics and metabolomics, to enable the application of synthetic biology to make building blocks for new polymer materials. An idea here is that we want to accelerate the discovery of bio-derived materials. We want to introduce new pathways into microbes such as yeast or E. coli to generate bio-derived monomers. We also want to introduce ways to directly polymerize these monomers in yeast to make bio-derived polymers. We want to have this to be beneficial to advanced material discovery to allow access to novel monomers, accelerate this design, build, test, learn cycle, and scalable synthesis of monomers and polymers. Shown here are just some examples that we proposed, which is to, based on the user's input, to perform mining, sourcing of the genes, building the pathway in these microorganisms, go through the design, build, test, learn cycle to generate different monomers that can be subject to polymerization to build different materials. Okay, so I think I'm right on time. So I want to acknowledge this center is just funded again by the NSF MIPS Award, and I look forward to working with everybody in the next 4 to maybe 9 years on different projects involving synthetic biology to make different compounds. Thank you very much for your time.
Okay, thanks a lot, Yi.